Bismillahirrahmanirrahim. We start the first ATP paper, which is an alternative to practical, and this is the one of the very latest ones, which is the November 2020, which was held during the pandemic, and of course, students were graded for it. So we're going to start. This is the first paper that I'm going to do in paper 6, which is an alternative to practical. Syllabus code is 5090. Of course, there is the other IGCSE, which we will be taking up later on. But at the moment, we're going to just do the paper 6 in 5090 O-level syllabus. As you all know, in the alternative to practical, which is the paper 6, this is syllabus 5090, and this is paper 6, and you have two variants, paper 6-1 and paper 6-2. The important thing to remember is this is a 1 hour exam. So you only have 60 minutes to do this exam, and you need to be a little fast about it. And you need to know how many marks there are for it. 40 marks. So you have a 40 marks paper, you have a 40 mark MCQ, and you have a 80 mark paper 2. So three exams to do. Paper 1 40 marks, paper 6 40 marks, and then the paper 2, which is 80 marks. Certain things which I want you to understand about an alternative to practical is that you are supposed to have done some practical work in the school, and if you haven't done it, even then you need to imagine that you're actually doing this in the lab, and what would be the conditions that would actually be needed in the lab, and what will be the steps that you will do. So always imagine yourself when you're doing a paper 6 is that you are actually doing this in the lab, and we are conducting this experiment under the laboratory conditions, and you'll be using all the apparatus which is present in the laboratory. Now question 1 says, when a seed germinates, the stem of the seedling grows upwards. A student wanted to investigate the effect of light and dark on the growth of seedlings. She grew some seedlings in the light and some in the dark for the same length of time. So if she left it for 3 days, all of them were left for 3 days. If she left them for 10 days, well, they all were left for 10 days. Now think you're going to do this in the lab, and you have these two pots, and you have a couple of seeds in one and a couple of seeds, the same type of seeds and the same number of seeds in both. And then one you've kept under a lamp, and you switch on the lamp for a specific period of time, and then you switch it off, while the other one you've kept in the dark and there's no light. So the photograph shows the seedling at the end of the investigation. So we see the ones in the light. What has happened? They've grown less tall, while the one in the dark have grown taller, and there are fewer leaves on the dark pot and more leaves on the light one. Then let's look at the question now. State two factors apart from light and dark that should be controlled when growing the seedlings for this investigation. Now, there are a lot of things which you can consider. You can consider, first of all, the water which you put in the soil, because this is a pot of plants, so it must have some soil. So you could have said water. You could have said temperature. If the temperature is kept the same, then of course the enzymes would work the same. So you could have said temperature, you could have said water. You could have said same size of the pot or same amount of the soil. So size of the pot should be the same. Then you could have said also same species. All the seeds must be the same species. You can't have in one pot something else and then the other pot something else. Then the number of seeds. So if you put 10 in one, then you should put 10 in the other as well. If you put 20, then you put 20 in the other. And then of course the next thing was carbon dioxide or oxygen. So whatever was provided to them, that area, because you see, you've got to realize is photosynthesis is going to start, and carbon dioxide is needed for photosynthesis and oxygen is needed for respiration. So you could have said either of them. So for 2 marks, you were sure to get, you just had to write 2 points. I'm not saying you have to write all these points. They're about 6 points, but you could have written any one of them. Then it says the student measured the length of the stem of some of the seedlings. One seedling grown in the light is shown in the diagram below. Measure the length of the stem between A and B. Of course, you just take a ruler and you measure the length, and of course, all you had to do was write that, and this was 23 millimeter. And as you go, Cambridge gives you plus minus 1, so you could have said 24 or you could have said 22. So this you had to measure, you had to use a ruler and you had to measure it and you had to just write that down. It was the units were given to you, didn't have to write the units. Please remember, we always use millimeter in these paper 6 exams. And let's look at part 3. The length of the stem in millimeters of 4 other seedlings grown in the light and 5 grown in the dark were recorded. So one was the one which we measured earlier, and then 4 other seedlings grown in the light and 5 grown in the dark were recorded by the student as shown below. So the dark ones are like this and the light ones are these. So this is the length of the stems. The length of the stems. Now keep concentrating, reading the question very carefully. Use this data and your measurement in A1 to complete the table below. It was basically very simple. You can see there are 3 marks. And what were the 3 marks for? Well, 3 marks was one for the 10 measurements entered. If you entered the 10 measurements, well, you got 1 mark for that. Then if you calculated the mean correctly, you got the second mark. So this was the mean for this one, and then this was the mean for this one. So if you calculated that correctly, well, you got the third mark. So 3 marks for entering this, and then of course calculating the mean. So it was basically a very easy question. I don't think it was difficult at all. And coming to the B part 1, another student thought that it would be better to measure 20 seedlings grown in the light and 20 grown in the dark. Suggest why this would improve the investigation. There's a very simple answer to that. It increases reliability. And I usually tell you to remember it is repeat is to increase the reliability. So this would increase the reliability. Reliability means that the readings are reliable. Now, what does that mean, readings are reliable? Say you took one reading and that is 10, something 10, anything. And the next reading is 20. And the next reading is 11. And then you get, say, 12. So you know now the trend is this. But then this one is an anomalous reading because it doesn't fit the trend. But if you only took one reading, then you won't know. So when you take more readings, like you take another reading and you take 5 readings. Usually, I always tell you that you should take 5 readings. So, if you take 5 readings, you know then this is the wrong one. So, you know the trend is this, and the next one is probably 12. So, you know this is the so you can do this one. So, you remove the anomalous readings, then you don't add the anomalous readings to it. So, increases reliability and or you can say easier to identify anomalies. Number one, increases reliability. Number two, easier to identify anomalies. Anomalies means that the readings that you got either by a mistake. We're not saying what mistake, human error, experimental error. We're not talking about that as yet. It's just an anomalous reading. Anomalous means just a mistake, an error. Error of judgment, maybe error you did something wrong in the experiment, maybe something wrong in the apparatus setting up, maybe just the way you counted something like bubbles per minute and you thought there were 10 but actually there were 15. So this is what we had to do for that. Now coming to question number 2, suggest why the growth observed in the dark might be an advantage to a plant in a shady forest environment. Yeah, well, if there's a plant growing in the shade and if it grows taller, then maybe it will reach more likely to find light. So more likely to find light, especially in a forest where there are dense trees. Then of course if it grows taller, then maybe there is some light coming and filtering through the foliage somewhere. So more likely to find light. Then part 3, use the photograph to describe one other visible difference between the seedlings grown in the light and the dark. Now, the previous diagram that we saw. So, what is the other difference that you can see? Well, you can see maybe in the light it is greener. It is greener in the light. Or you could have said something else like more developed, larger. So these are the wordings that you could have used. Either you could have said this is only 1 mark for it, but of course see that you do get your 1 mark. So greener, darker, chlorophyll present. If you're talking about the light one in light, you must tell me what you're talking about. Are you talking about the light or the dark between the seedlings? Because light has been mentioned first in the question, so we're usually talking about that which is mentioned first in the question. Coming to the C part of the question, some scientists wanted to investigate the effect of fertilizer on the growth of wheat. Wheat seeds were planted in soil to which nitrogen fertilizer had been added. After the seedlings had grown to a height of 8 centimeters, they were measured at regular intervals. The mean heights of these plants are shown in the table. So time of the day 0, mean height of the plants was 8 centimeter. Then on the 10th day it was 10 centimeter. Then you can see on the 30th day it was 29, then 60th day it was 58, then 90th day 73, and by 110 days it was still 73. So it didn't grow any taller after the 90th day. Maybe it stopped growing on the 95th day or the 96th day or whatever, but then after about 20 days when we did see the height, it remained the same. So we have 0 first, then 10, then we have 30. So then we have a difference of 20 days, and then after 30 then we have a difference of 30 days, and then 30 days, and then again 30 days. So it's a little variable that you know that 20 days it was here, and here it is 30, and here it is 30, and here it is 30. So do look at a table very carefully and try to figure out where there is some sort of a difference and which doesn't tally with the sort of normal pattern. Then it says construct a line graph. So you have to do a line graph of the data on the grid below. Join your points with ruled straight lines. Now this was specified this year. Otherwise, it just says join a line graph, and you know you can either do it with a ruler, point to point join with a ruler, or you can just do a curve through it, through the points. The rule which I want you to remember is that the first column in this table, this is the first column in this table. Now the first column in the table is always, the first column in the table is always the independent variable, and it is always plotted on the x-axis. That is a mnemonic for it. Fix. First column is always the independent variable. Please remember there are two variables that we talk about, dependent and independent variable, and that is always plotted on the x-axis. And the other one, which is the dependent variable, is always plotted on the y-axis, and that is the result, what you are going to be measuring, bubbles per minute or the length of the plants or the height of the plants, and that is always plotted on the y-axis. So this is the normal rule is the second one will be the one which is going to be plotted. So that is the mnemonic is dependent variable is always the result which you are calculating in the lab or measuring in the lab, and that's always on the y-axis. Rule is that you always write this whole thing. So mean height of plants centimeter should come on the y-axis. Mean height of plants centimeter must come on the y-axis. And then this thing, whatever it is written, please no abbreviation. Time slash days must come on the x-axis. So this is how I want you to remember this, is that you have to have the entire heading. Please do not make any abbreviations. Do not add or subtract anything. So time of the days. Now, if you've just done this, if you've just labeled the axis, you get 1 mark for labeling the axis. Mean height of plants is on the y-axis and x-axis as time and days. You haven't drawn any graph. You will still get 1 mark. Now, how do you decide the scale? You see, it's got 0 to 110. So you look at the smallest figure and then you go to the biggest figure. Sometimes, it could be the smallest figure could be 50 and the largest figure could be 110. So then you have to spread it between 50 and 110. You don't have to start from 0. But here, because we have a 0, so we will start from 0. So we will ensure that 110 comes somewhere here, right at the end of the graph, at the end of the grid, at the end of the graph paper. So this is how I decided to take 0, 20, 40. I mean, you shouldn't be just using a small portion of the graph. You should be using two-thirds of the entire graph. So please remember is that you have to spread out the, how do you have to spread out the graph, and you have to get a graph which fits this entire graph paper. Now let's look at the y-axis. On the y-axis we have the smallest figure is 8 and the largest is 73. So let's see how we can spread this out in on the y-axis, 73 and 8. So we've got to spread this out. If you look at how I've spread this now, I have put 0, and for every 10 boxes I've said 15, 30, 45, 60 and 75. But this becomes a little difficult, the scale. The scale becomes a little weird. For 10 small scales we have 15. For at 0 I have to plot an 8, and I'm going to be rather confused about it. How am I going to plot an 8? So you could have done this, but you could have done another way of another scale also, which is slightly, but this was also acceptable. You could have done this scale too. And for every 10 small squares of 20, 40, 60, 80 and 100. Because 73 is somewhere else. So you are using nearly two-thirds of the graph. Two-thirds of the graph paper given to you. Now you can see I've plotted the points, but there is something wrong that I've done. I haven't circled them. You see, because my pen is a little, so you have circled them. You must circle all the points. You must first plot the points and then you must circle all the points. That's one thing which they expect us to do in the graph. And then now you have to join them. So if you plotted, now the first mark was time on the x-axis and axis fully labeled with units. Then linear scales and values at origin and occupying at least half the grid. This got you the second mark for you deciding to what scale you're going to use. Then plots, 6 points plotted correctly. And then of course, rule lines between points and no extrapolation. Extrapolation means you don't extend it. You don't extend it in any direction. So this is what we had to understand is that you will not, so now joining it with the ruler point to point. I might not do a good job at it, but I'll try my best. So not a very neat job, and here you can see there's a mistake here. So joining it, rule lines between points and no extrapolation. Extrapolation means that you do not extend this. You do not extend it. That was the very important thing, is that you do not extend it, and you do not extend this to 0 on this side either. You don't extend this to 0 on this point here, or you don't extend it here. That is called no extrapolation. Whatever points are given, you just join those, and that is it. So this is how you had to plot a graph. So there are 4 marks for a graph, and I feel is no student should be actually losing marks in this graph, because labeling the axis, choosing a scale, then plotting the points, and then joining them is a basic thing that you've done in maths many times, you've done in physics as well, and I'm sure you can all do it. But if you just put your mind to it, I'm sure you can all do it. It says here, use your graph, find the mean height of the plants at 50 days. Now all you have to draw is you have to take, see where it was the 50 days. You have to draw a line like this, and then you have to go here and you have to read this off. And this should have been, actually it's 48 plus minus. So you must write the unit here, because this was the unit which was given here. So 48 centimeter, and it was a plus minus 1, so you could have said 49 or 47. And why were there 2 marks for it? Because there is 1 mark for this 48 centimeter, and 1 mark is for the fact that you showed me this line on the graph, which of course not such a thick line, but of course a thin line, and you showed this to me on the graph. So that was because it says show your working, show your working on the graph. So please remember to do that. Read the question carefully. Do not miss out a mark for this. As you look at part 3, describe how the rate of growth of these plants change between days 0 and 110. So you have to look at the graph and you have to give me the statistics from the graph. So you see there is this very less growth here, and then we see this sharp increase here, and then we again see here that there is no change. So you have to look at the graph and give me pointers from the graph. And if you look at what the answer says is growth slows till day 10. Growth is slow till day 10 and increase between days 10 and 60. Then increase and then decrease between days 60 and 90. So, and the last point was between day 90 and 110 growth stopped because the length did not increase. So these were the three marks and these were the three. So whenever it's a describe, it's just a describe means no biology, and you have to just give me points from the graph, and you just have to look at the graph and tell me what is the graph telling you. So describe how the rate of growth of these plants change between day 0 and 110. Then coming to part 4 of the question, describe how to complete this investigation to find whether fertilizer does have an effect on the rate of growth. So we would have two types of experiments. In one, you would have the fertilizer, in the other, you won't have the fertilizer, so no fertilizer. And the same type of wheat seeds and the same water supply, so that the only difference we have is that one crop has the fertilizer and one does not have the fertilizer. Then coming to part 5, increase in height was used as a measure of growth. We said, okay, we planted the seed started to grow. So this was the height. And then after some time it's grown a little, so increase in height. So increase in height was used as a measure of growth. So just two other plant features that could have been used to measure growth. Yeah, if the number of leaves increased, now there were 5 leaves, now there are 6, now there are 8, now there are 9, now there are 10. Or if we could see the surface area of the leaves, because you see the number of leaves might say the same, but the leaf which was this big now is becoming bigger, then is becoming bigger. So the leaf size is changing, which means the surface area of the leaf is changing. So number 1, number of leaves, number 2, surface area of the leaves, and number 3 is the dry mass or the yield of seeds produced or the plant. Now dry mass is a very good measure because it tells us actual organic matter. Because dry mass means there's no water in it, but dry mass is the actual cell matter like cell wall, cell membrane, and the organelles, but we've removed the water. So dry mass is a very good measure of growth because more the number of cells in any organism means there has been more cellular matter. Now that finishes this question, and this was for 22 marks, and you know the 40 mark paper, this was 22 marks, so nearly 50% of it was examined here. And then we have another question on this, which is, of course, I'm going to do, there are two more questions which I'm going to do in a second video, which will follow after this video. And thank you for now.